Cool. We could probably go ahead and get started. Hey, folks. Happy Monday. How's everyone doing? How's life? Good. Perfect. Perfect. So in terms of, oh, geez, what did I do? Why did you end up over there? So in terms of logistical announcements, it's week 7, so midterm is officially over. So people that took it 2 weeks ago are probably like, what? Why is this still going on? But yeah, so congrats everyone. You're done with the midterm. I posted the grades today, but there shouldn't be any surprises because it should be exactly what you saw in Prairie Learn right after you took the exam. So I don't think Prairie Learn has a way of showing you what the right answer is, but if you got any questions wrong, it shows you what questions you got wrong and what answer you put. So feel free to come to lab section, like ask the core staff or like any help on figuring out any of those questions. Yeah, so I guess basically next thing is, oh, and I forgot to post the problems for let me pull that up real quick. CSE 185. So lab 7 is released, and I forgot to put the non-coding practice problems for lab 7. I think, yeah, but I'll put them probably later today or whenever I remember. But just like with the midterm, the final is going to be the same deal. No surprises. Every single question from the final is going to come from these practice problems, which are going to come from the non-coding problems from any of the 9 labs. So midterm was 1 through 4. Final is comprehensive 1 through 9. But every single problem, again, is going to be from those problems. So if there's anything that you missed, please come to lab section, study those specific problems. That's the best way to improve your grade, is just make sure that anything that you missed, you know exactly how to solve it. Are people able to schedule their finals yet? So the finals also going to be through Triton Testing Center. So people are nodding. So if you haven't scheduled your finals yet, like please schedule your final ASAP to make sure that you get a good spot. But same logistics as the midterm. I believe it's just at the Triton Testing Center again. I think, is it weeks 10 and finals week? That's what I'd asked them, but I don't. Oh, it is, yeah. So if you want to take your final during week 10, you can. If you want to take it during finals week, you can. Week 10 won't cover any new stuff that's going to be testable on the final. It's just going to be like, let's teach you how to get stuff set up on your own computer and kind of like some last thoughts and kind of just finish up the course, study for the final, all that good stuff. So yeah, so if you want to like leave campus early for finals week, like definitely feel free to take advantage of those week 10 times. Cool. So yeah, so I'm gonna, if I remember after class, I'm gonna post the lab 7 non-coding problems here, but regardless, you have access to them because lab 7 is released. And yeah, any logistical questions, concerns? Yeah, go for it.
Just wondering, for after the class, is this going to be like archived so you can access the information later?
Oh, good question. So the question was, is this going to be archived after the class? So Prairie Learn is going to lock down after the class. So definitely back up any code, any notes that you want from Prairie Learn. Canvas, you should still have access to. So on Canvas, I have some helpful resources of like, let me bust out Canvas real quick. CSE 185, go to student view. So on Canvas, I have this, this tools list that the TA and tutors helped me write that just kind of comprehensively lists all the tools that we covered in this class with like brief summaries of like what each category of tools does, as well as some that we didn't cover in class, but I wanted to, but it would have been too much work. So I think this is probably the key takeaway from this class, and you'll have access to this indefinitely. But yeah, any code that you wrote for the labs, definitely back all that stuff up before the quarter ends. Yeah, great question. Any other questions about logistics, concerns, comments? Everyone's all good? Awesome. Perfect. Okay. Any questions on Zoom? Zoom people are good? Perfect. Okay. So let me leave student view and then close out of this. Okay, so let's first quickly recap everything that we've talked about so far. So we introduced this class week 1 by introducing all these cool real world problems that we want to tackle. Week 2, we talked about de novo genome assembly and de novo genome annotation. Week 3, we said, okay, now that we have our high quality genome, if I sequence another sample from the same species, how can I use that reference genome to help me? So we talked about read mapping. Week 4, we talked about consensus sequence calling and variant calling. So basically, now that I'm sequencing all these samples, how do I reconstruct their consensus genome sequences or their variants, their differences from the reference genome, as efficiently as possible? Week 5, we talked about multiple sequence alignment. So now that I have millions of genome sequences collected from all over the world, how do I line them up? And remember, the goal of that was to determine sequence level homology. So basically, in the multiple sequence alignment, each column corresponds to one specific position of the ancestral genome sequence. So even if individual sequences differ in what letter they have at that column, they all theoretically evolved from the same exact letter in the common ancestor. Either they were inherited directly or they mutated, but from that specific letter. So that sequence level homology was useful because then in week 6, we talked about phylogenetic inference. So phylogenetic inference used the positions of our multiple sequence alignment in order to infer a phylogeny. And we specifically focused on what was called maximum likelihood phylogenetic inference, where we basically said, here's this rapid fire of models of substitution that you don't have to memorize the details of each one, but just know these are the types of assumptions that each of them makes. Given these models, we can come up with a way of scoring the likelihood of observing a given set of sequences given a tree and the model parameters. And that score is effectively used for some type of a tree search where we create a quick and dirty tree, we modify it, keep scoring, modify, score until we eventually converge on a tree that maximizes that likelihood score. Okay, and that's where we kind of left off. And what did I mention about the trees that we get from those models? Unrooted trees, exactly. So the models that we were using are what we call time reversible. So the mutation from nucleotide X to nucleotide Y is the same process as the mutation from nucleotide Y to nucleotide X. So we don't know any directionality of mutation. We just know how many mutations happen. So the trees that we get, they're still informative. We know the topology, we know the branch lengths, but we don't know the root. So we don't know where the common ancestor is of all these sequences. Okay. Questions about any of that rapid fire that I just summarized real quick? Everyone's with me? We're almost done with our journey. 2 more weeks of new stuff. We got this. 2 more or 3 more weeks of new stuff. I'm getting ahead of myself. Okay. So today, now we're going to take that unrooted tree that we got last week, and we're going to talk about tree rooting and tree dating. So specifically, here's an outline of what today's topic is going to be. I mentioned the trees that we got last week are unrooted trees. We're going to talk about a few different approaches that people use to determine where is the root of this tree. So specifically, we're going to talk about outgroup rooting. We're going to talk about midpoint rooting, and we're going to introduce this concept known as the molecular clock and talk about how that's going to help us both root and date the tree. What does dating a tree mean? Don't worry about it yet. We'll talk about it when we get to the 4th bullet. But any questions before I jump in? Folks are with me? Awesome. Okay, so let's jump in. So first, rooting of phylogeny. So remember, the evolutionary models that we use to infer phylogenies were time reversible. And because the rate from X to Y is the same as the rate from Y to X, we can't infer the directionality of evolution. So the trees that we get are unrooted trees. But we want to be able to infer the most recent common ancestor of the tree. If we can figure out where on this tree was the most recent common ancestor, we can then reroute the tree, and that tells us the evolutionary history of these sequences in time, not just distances from each other and closeness, but actually this event happened first, this event happened second, this event happened third. And where this root exists can be very informative to people that are studying like the origin of a new pathogen, for example, COVID origins or HIV origins or Ebola origins or insert whatever virus people are trying to find the origins of. This is precisely how we find those origins. Okay, so our overarching question for the majority of this set of lecture slides is how do we find the root of our phylogeny? So again, I'm going to summarize the computational problem. Input is an unrooted tree. Output is determining the exact position, so what branch and where along that branch is the root of this tree. Okay. So folks are clear on the computational problem we're tackling? Awesome. Okay, let's jump in. So the first technique that we're going to talk about, which in my opinion is typically the simplest to understand, or at least the simplest and most reasonable to actually use, is called outgroup rooting. So remember, kind of a priori, we're assuming that all modern organisms evolved from some common ancestor billions of years ago, and we have this crazy, crazy, crazy big tree of life. And remember, speciation is the process in which a single ancestral species diverges into 2 distinct species. So the parent species splits into 2 distinct child species. every single sample in the phylogeny that we constructed back in week 6 has a most recent, oh, and by the way, I don't know if I'm using this terminology that maybe I didn't remember to define, but MRCA stands for most recent common ancestor. That's the root of the tree. It's the most recent common ancestor of your collection of sequences. If you want to find the most recent common ancestor of a subset of your sequences, it's just the root of the subtree that includes all of them. So most recent common ancestor of a collection of sequences is just what is the node that all of these are descendants below. But yeah, so all of the samples in the tree that we built in week 6, even though that was an unrooted tree, in this real tree of life, they have an MRCA that exists somewhere here. Okay. So what if in advance, before we do our analysis, we know that there's some sample that diverged earlier in time than all the samples in our data set. So for example, we know that like with SARS-CoV-2, we know that if there's some SARS-like coronavirus that's not SARS-CoV-2, maybe a bat virus that's similar to SARS-CoV-2, that must have split from the common ancestor of the SARS-CoV-2 samples kind of like earlier in time. So there was some common ancestor. That one branch gave rise to all of SARS-CoV-2. The other branch gave rise to this SARS-like bat coronavirus that's not directly related, like it's not a subtree of SARS-CoV-2. If we're thinking about like humans, maybe out of all these human samples, well, chimpanzee evolved and like branched out before the common ancestor of all humans. So if I want to find the most recent common ancestor of a collection of human samples, I could stick in a chimpanzee sample, which must have diverged earlier than any of the human samples. So these terms that I'm defining, this like SARS-like coronavirus when I'm studying just SARS-CoV-2, or like a chimpanzee sample when I'm describing human samples, this is called an outgroup. This is a group that we know in advance is outside of the subtree that all of my samples exist in. And we know this biologically just based on prior knowledge. So when we do a phylogenetic inference, what we could do is throw in that outgroup sample, infer the evolutionary tree using the exact same techniques we talked about last week, and wherever that outgroup hits the rest of the tree, that point is the root of the rest of our tree. So as a visual, let's imagine we have that same unrooted tree that I talked about earlier where A, B, C, and D are all in the same species. But now what happens if I just mix in an outgroup sequence or maybe multiple outgroup sequences? So here I'm drawing it as if it's just one sequence, but I could drop in like multiple sequences. So like if I'm studying SARS-CoV-2, I could stick in multiple bat SARS-like viruses or maybe multiple SARS-1 viruses. But yeah, basically the outgroups, they would still be part of this evolutionary tree, but there would be like a really, 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 really long branch that goes to them because they've diverged way further back in time. Okay, so first, does this figure kind of make sense? Is it intuitive why including them would be like way off on this tree? Okay. So I don't know the root of this tree, but I don't care. Where this branch intersects the rest of the tree, which is what I actually care about, that point is the root. The root of this original tree is probably somewhere along this really, 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 really long branch, but I don't really care about the outgroup. I just care about my samples, and the outgroup was just a way of determining, oh, hey, this point is the root of this tree. Okay. So does the idea make sense? Questions? Okay, so anytime I introduce a bunch of different techniques, I'm always going to have the pros and cons. So what are the pros and cons of outgroup rooting? So one pro is that it's super computationally easy to do if we have a reasonable outgroup. I just run my exact same analysis, just with maybe like one additional sequence or maybe a couple additional sequences if I want multiple outgroups, and it just works out of the box. Find the branch that it intersects, output the tree rooted at that branch. So it's linear time once I finish my analysis. Really easy. Oh, there's a question in the chat. Based on B, C, and D being equal distance from each other in the first tree, is making B higher up on the second tree arbitrary? Let me see. So the question was, B, C, and D. So actually B, C, and D are not equidistant from each other. So in this example tree, I'm just kind of arbitrarily drawing C and D as being like this branch length is the same length as this branch length. So when I root somewhere further to the left of this tree, they're going to fall right next to each other equidistant. But B is actually slightly higher up. And the reason for this is because, oh, this is a bad drawing. Whoops. There's supposed to be a branch. Good catch. This is a mistake. I think my later slides fixed it. Let me see. Spoilers. Oh, geez. Oh, no, I didn't fix it. Whoops. Good catch. This is actually a bug in my slides. I'm going to fix it real quick. And I'll do it on camera so everyone can see. So basically, I drew this as an X because it was easy to draw, but there's actually supposed to be, oh, I think in the next set of slides I fixed it. Whoops. So there's actually a branch here in between the ancestor of like the intersection of A and B versus the intersection of C and D that I forgot to include. So there should be a horizontal branch here. Let me real quick move my screen over. Sorry, good catch, because this actually, it doesn't change the big picture, but it is important for the two figures to match up. Whoops. I think it was, did I fix it in the next set of slides? Sorry. Nope. Okay. I'm just going to fix it right now. Okay. So what we're going to do, I'm gonna ungroup these. Oh, I even got it ready to fix. Whoops. Oh, geez. There we go. And I don't want this one. I don't. Oh. Darn. Okay, I'll fix it later. I'm gonna waste too much time in class. So I'll just leave myself a reminder to fix it. So yeah, sorry. So the correction should be, there should be a horizontal branch here. So it should be A, B, and then a branch, and then C and D. And that branch that's missing is this branch right here. So good catch. Okay, sorry about that. So that's a detour. I'll fix that, and then I'll kind of update the slides. So yeah, so the pros and cons of outgroup rooting, it's super computationally easy. And the other pro is it's actually very, very, very accurate if we have a reasonable outgroup that we can choose. A con is that if you pick a bad outgroup, it's going to really screw up the whole process. So what are some ways that you can screw up? Well, some of the higher taxonomic groups in the tree of life might not have an agreed upon outgroup. So things that are like way back in time in history, we might not know where some of those branches fall. There's actually some, let me pull up one that was like a big thing in my PhD that we ended up still not solving. Let's see. There we go. So I'm going to show this little cute animal. Super cute. The tree shrew. So I'm going to ask the audience a question. Is this a rodent or is this a primate? Let's think. Is it a rodent or is it a primate? So rodents are like mice, rats, yada, yada. Primates are like chimpanzee, human. Yeah, go for it. Oh, ooh, does the tail have fur on it? Yeah, it does. There we go. Here's like a tree shrew. Oh, that's sad. This one's dead. I want this live one. Any thoughts? It's all good. The uncertainty here is exactly what the scientific community has as well. So in terms of morphological features, historically, the tree shrew was lumped in with rodents. But when people started looking at its genetic signature, genetically, it seems like it's under the primate family. But still, it's kind of an unresolved consensus of like, is this on the rodent side or is it on the primate side? So if I was doing an evolutionary tree of a bunch of tree shrews and I wanted to throw in an outgroup, well, I guess I could have used some rodent or some primate, because I guess either of those would be outgroups of them in general. But like if I was doing an evolutionary tree of rodents and I wanted to include this, that might screw up my results. Or if I want to do an evolutionary tree of primates, if it actually isn't a primate, that might screw up my results as well. So where tree shrew falls on the tree of life is kind of a big open question. Or big is, I guess, relative. I don't think the general public actually cares that much, but it's an uncertain thing in the evolutionary biology world. Did you ask if it has fur? Because monkeys and stuff have fur. I think that's actually something that is a morphological thing that should have been caught as evidence for primates. So my people, our team, this is a primate because we're the genetics people. Oh yeah, that's true. Our squirrels were, yeah, I don't know. Our squirrels rodents? Squirrels?
Squirrels are rodent, right?
I don't know. Squirrel. They're oh yeah, they're smaller, medium-sized rodents. So oh, capybara. Where'd that go? Where did my capybara go? So that's true. Yeah, so actually the tail, maybe not. It's a mystery. That's true. That's true. They do have fluffy tails. So it's not always clear. This assumption that we can just trivially pick an outgroup, depending on the research question. So if we're only looking at one species, sure, it's easy, pick something that's outside of that species. But if it's not that species, like if it's a mixture of different species that we're trying to find the evolutionary history of like a bunch of different species, then the outgroup might not be super clear. Another problem is even if we do pick something that's correctly an outgroup, like you might say, oh, well, these are all multicellular organisms. Let me stick a bacterial species as an outgroup. But the problem is if it's too diverged, the evolutionary tree is going to get kind of screwed up. And it turns out that if you have super, 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 super long branches on a tree, it's hard to infer those branches accurately. The reasons for that I can actually kind of briefly get into. If you kind of imagine, and I'm not going to test you on this, but most phylogenetic methods assume what's called the infinite sites assumption, which assumes that the length of the genome is large enough that we can kind of assume that it's infinity. So what that implies is the same position of your genome probably doesn't get mutated multiple times. That's not actually a realistic assumption. Even with SARS-CoV-2, we've seen multiple individual lineages of COVID like independently mutate the same mutations, but it's a reasonable assumption for most of the genome. So if we theoretically plotted on the horizontal axis number of mutations between two sequences, and on the vertical axis, we plotted the Hamming distance between two sequences. This curve would actually start fairly linearly at the beginning. If you don't have a lot of, and people should hopefully remember Hamming distance. I think I had you implement it earlier, but Hamming distance is number of mismatches between two sequences. And in this case, I'm going to assume proportion mismatches, just like normalized by the length of the alignment. So when you have relatively few mutations, they should hopefully all hit different positions of the genome. So you're just going to have like a fairly linear relationship. But then as you grow, it's going to start flattening out as you start hitting the same site multiple times, which is a violation of the infinite sites assumption that most things assume. So I'm going to ask not a trick question, but a CSE 21 question. If I'm doing Hamming distance as a percent, what is this line that it converges to? Is it 50? So let's think about this. So this is the Hamming distance. Let's assume mutations are infinite. So we've mutated literally every single position an infinite number of times. So what would be the Hamming distance between these two sequences? And I'll rephrase. Oh yeah, go for it. Is it, sorry? Is it the length of the genome? So 100%? So I have one guess for 50%, one guess for 100%. So I'm going to rephrase the problem slightly to sound more like CSE 21. So what is the expected Hamming distance between two random sequences? So let's think one position. What is the probability that one position is different between two random sequences? So I randomly sample a letter ACGT, randomly sample a letter ACGT. What is the probability that these two letters are different? Yeah, go for it. 3/4. Why 3/4? Exactly. So I have 4 options. I randomly picked one of them for this one. That's equivalent to me saying, okay, I picked one for this sequence. What is the probability that the random one I picked for this sequence was a different one? So it has a 25% chance of being the same one, 75% chance of being one of the other 3. Now do that across the entire sequence. So I'm flipping a coin with 75% chance of heads for each position of the genome. Overall, the expected Hamming distance, the expected proportion of positions that'll be different, is going to converge to 75%. That's just a fun little combinatorics problem. But what am I getting at here is that if the outgroup is too distant, we're going to hit some sites multiple times and we're going to underestimate the actual number of mutations. Because like it's indistinguishable to be able to, like, there are some ways of correcting the estimated number of mutations from Hamming distance. Like if we're over here, maybe I can do a correction, but like here versus here, it's impossible to tell these two things apart in terms of just Hamming distance. Okay, so the key takeaway there is if my outgroup is too diverged, it'll screw up the entire phylogenetic inference process. So my entire tree sucks, not just the root. So yeah, and then it can also mess up the multiple sequence alignment, because if we're getting a bunch of random mutations at every single position, even the multiple sequence alignment might accidentally put things together just randomly by chance, even though they were two separate things that just mutated to be the same thing. There's also a concept that I'm not going to cover in this class, but if you ever work on phylogenetics, this is a useful key term to look up. It's called long branch attraction. This is a problem where if you have a really long branch in an evolutionary tree next to a really short branch on an evolutionary tree, the topology of which branch goes with what is very hard to estimate. So you get topological inaccuracies when you have a really long branch next to a really short branch, just because of mathematical properties that you don't have to worry about. But if you ever work in this in the future, this is a useful thing to look up later on. Anyway, I'm not going to test you in here. Okay. So outgroup rooting, great option, especially for SARS-CoV-2. That's great because we have plenty of reasonable, relatively closely related outgroups. We have a bunch of SARS-like bat viruses. We have SARS-CoV-1, a bunch of other coronaviruses. We have good outgroups for SARS-CoV-2. Okay, any questions? That was my last slide on outgroup rooting. Questions, concerns, comments? Kind of makes sense. Okay, so this, if you can use it, if you have a reasonable outgroup, this is typically the preferred option. Another option is what's called midpoint rooting. So for midpoint rooting, let's see. What if, so I'm going to start with the assumption that we're going to be thinking about that then motivates midgroup rooting. So the assumption before we talk about midpoint rooting is, what if the true rooted tree was ultrametric? So ultrametric is a keyword that just means every single leaf is exactly the same distance from the root. So what if the real tree was actually ultrametric? Well, the longest path in the tree is a path from a leaf on one side of the root to a leaf on the other side of the root. Doesn't matter which leaf. In this case, I've arbitrarily chosen A and D, but it could have been A to C. It could have been A to B. As long as it's A with one of these 3, all of those are the exact same total distance because all of these are equidistant from the root. So it's distance from one of them to the root plus distance to the other one, which is just 2 times distance from the root. So this motivates a very trivial rooting option where I could just take the longest path and pick the midpoint of that path as the midpoint. So midpoint rooting just says root the tree at the midpoint of the longest path between two leaves. So calculate all pairwise distances, take the longest one, find that path, and just stick the root right in the middle of that path. Okay, is the idea clear? Is the assumption that we're making clear? Questions kind of make sense? Okay, as always, pros and cons. So pro, again, this is super computationally easy. I could do this. If you do all pairwise calculations, it's technically going to be quadratic time, but you don't even have to do all pairwise calculations. I can find the longest pairwise distance in linear time by just rooting arbitrarily and then doing a post-order traversal. Don't worry about how that works. I'm going to show you a tool that I developed next week that does exactly this, but you can actually do this in linear time. So it's super computationally easy. The other benefit is, unlike outgroup rooting, where before we do the rooting, we have to have a reasonable outgroup. I don't need any additional information. I just throw the sequences from my collection into the tree inference process, take the longest path, root it there, no additional information needed. Okay, so for the cons, I'm going to first ask kind of a theoretical question. So this assumption that every single leaf is equidistant from the root, is that a reasonable assumption? Okay, people are shaking their heads no. Why? Other than the fact that my tone is kind of alluding to that. Are there any like real world examples that you can think of where equidistant from the root is a bad assumption? Any thoughts? Think about COVID. So what if I wanted to make an evolutionary tree of COVID sequences? What are the leaves? Any thoughts? Yeah, go for it. So each leaf is like one sample that I collected. So one person does a nasal swab, I sequence that sample on that specific day. This is one leaf in my tree, right? Because I get one genome sequence and the genomes are the leaves of my evolutionary tree. So we've been sequencing throughout the entirety of the COVID pandemic. So I have some sequences collected back in like December 2019. I have some sequences collected in like, I forgot when the Diamond Princess had landed in San Diego Harbor. Was it like February 2020 or something like that? We got those sequences. We got like when it was introduced in March 2020. We have like Return to Learn was sequencing on UCSD campus throughout the pandemic. So the true evolutionary tree very significantly deviates from ultrametricity. I have some sequences that were collected. So if the ancestor is like November 2019, I have some sequences collected December 2019, some sequences collected January 2020, some sequences collected February 2020, some sequences collected 2025 because COVID is still around. So the tree is very much not ultrametric. We have some very early sequences, some very late sequences. So it deviates very significantly from ultrametricity. Not all the leaves are the same distance because they weren't all sequenced on the same day. So yeah, so with viruses, the trees are actually by definition not ultrametric because I'm sequencing over time. So the later samples are going to be further from the root than the earlier samples. So in the case of viruses, this is actually a very bad approach unless for some reason I'm guaranteed all of these samples were collected the exact same day. And then even with ultrametric sampling time, so even if I sequence everybody in the world on the exact same day, the mutation rates are not necessarily constant. So even if the times are the exact same time point, the number of mutations from the root might differ across each of them because the mutation rates are going to be slightly different. So yeah, very bad assumption. It's very computationally easy. I don't need an outgroup, but it's terrible assumptions. And it's going to get you really wonky results on like real virus trees over multiple years. Okay, questions? Does that rationale kind of make sense? Yeah, go for it, go for it.
When is this type of method ever used?
Ooh, when is this type of method ever used? So one common problem that people want to do is like if we're studying the evolution of humans, so there's like biblical origins for the terminology, but there's this concept known as like Y chromosome Adam and like mitochondrial Eve. So Y chromosomes are So all your other chromosomes, you have 2 copies, one from mom, one from dad, but like within your cell, you don't know which one is from which parent. You could do some genetics to figure out using your parents' genetic sequences, like what it is, but it's not very clear. But your mitochondria, you only inherit from your mom. So mother to child, female child to their child, female child to their child. We have a straight lineage of mitochondrial DNA that goes along maternal lineages. And your Y chromosome, if you're a male, you only inherit Y chromosome from your father. So Y chromosome lineage goes all the way down. So we have a non-branching, unambiguous path of just linear branching, like linear inheritance of Y chromosome. So one question that's very commonly asked is like, when did humans exist? Like, when did humans branch off from their common ancestor, or like the out of Africa theory? Like, how did humans come to like spread across the world? And that has a lot to do with when is the common ancestor of all human sequences. So humans have existed for a very long time. So even though there is some slight deviation in like, if I collect a sequence from someone in 2000 versus like 2010 versus 2020, our common ancestor is like way back in time. So these slight differences in the dates are like negligible compared to the true pairwise distance. So for problems like that, midpoint routing can actually be reasonable because the common ancestor is so far back in time that like the deviation of collection times is negligible in comparison. Does that kind of make sense? So it is useful in some context, but the context that I set up this class with like viruses as the focus is a terrible context. But in other contexts, it is actually, you just have to convince yourself, are my trees going to be close enough to ultrametric? Yeah, great question. Any other questions, concerns? Yeah, go for it, go for it.
So is this the only like method if you don't have an outgroup, or are there other ones that are maybe more complex?
Ooh, are there other methods if you don't have an outgroup? Great question. So we have one more method that's more accurate typically in practice, but is trickier and requires a bit more math. But I'll pause before I do that. Any other questions about midpoint routing or outgroup routing? And there are even other ones that I didn't include. So like someone from my PhD cohort created a method called like min-var routing that's trying to minimize the variance in the root to tip distances. All sorts of stuff, but they all kind of are predicated with what are some assumptions we can make about how the tree should look like. Now let's optimize for that property, whatever it is. Great question. Okay, so the last section of this talk, I'm going to be talking about what's called routing, routing via the molecular clock, and then also I'm going to introduce a concept known as phylogenetic dating. So first, what is dating a phylogeny? When we inferred a phylogeny last week, the branches were in unit of number of mutations. It was actually expected number of per site mutations, so it was normalizing by the length of the genome, and it was averaging across all the positions, but still some metric of number of mutations. So the trees that we get, the branch lengths represent how many mutations occurred from parent to child. But it would be super useful if instead of having the branches in unit of mutations, we could have the branch lengths in unit of time. So in other words, how much time has passed from parent to child. So if I'm studying an epidemic, temporal information of like, this is how much time has passed is very helpful. So just given sequences, so build a multiple sequence alignment in week 5, use maximum phylogenetic inference in week 6, I can infer a mutation phylogeny, so a phylogeny where the branch lengths are in unit of mutations, just using my sequences. But what if I have additional temporal information? So for example, if I'm talking about COVID-19, each of those samples that I've collected came into the clinic on a specific day, got their nasal swab, and then I sequenced it. So I theoretically have sample collection dates for each of the leaves of my tree. So what we can do is if we incorporate this temporal information, we can scale the branches of our tree so that instead of in unit of mutations, they're in unit of time. Whoops. And this is mathematically equivalent to estimating mutation rates, so number of mutations per time for each of the branches of our tree, and then scaling it accordingly. So if the branches are a number of mutations and I estimate mutations per time, I can just divide number of mutations by mutations per time to get time. So scaling the branches, this is equivalent to estimating mutation rates for each branch. Okay. So how do we do this? I'm going to pause for a second. Just, is it clear that estimating mutation rates for each branch is mathematically equivalent to scaling it from mutations to time? Okay. Okay. So how do I actually estimate those mutation rates? Well, there's this concept in evolutionary biology known as the molecular clock. And this molecular clock, it's an assumption that we're saying mutation rate stays roughly constant over time. It's going to have some variation. Maybe some branches are going to have a higher mutation rate, some branches are going to have a lower mutation rate, but over time it should stay roughly constant. So what we could do is, if you gave me a rooted mutation tree, so I'm going to assume for a second that we already have found a potential root. What I could do is I could plot the root to tip distances on the vertical axis versus the sample date on the horizontal axis. So in other words, horizontal axis is the sample date, for example, what year or what day or whatever time unit that I'm looking at. Vertical axis is the root to tip distance, so sum of the branch lengths from that leaf to the root, just add up all the branch lengths. And each of these dots would be one leaf in my tree, where the horizontal coordinate is the time that I collected it. The vertical coordinate would be the sum of the branch lengths from that leaf to the root in my rooted mutation tree. Okay, so when I show this, what does your eye automatically want to do? Yeah, I see people like doing exactly. We draw a line, and what does this line imply? Well, human eye likes drawing lines, but remember, under this molecular clock assumption, we're assuming mutation rate, so mutations per time, so slope of a line, is staying roughly constant over time around some mean. So if we draw a best fit linear regression connecting these points, the slope of this line is the mutation rate. It's the average, or we can assume that it's going to be like the average or the best estimate of the overall mutation rate. It's number of mutations over time. And what we could do is, okay, well, what is the time of the most recent common ancestor? We call this TMRCA, time of the most recent common ancestor. That's the x point when the y coordinate is 0. So when I'm 0 distance from the root, what is the date? Okay, so we can figure out the slope, which gives us our mutation rate, and we can find the x-intercept, which gives us the date at the root of the tree, so the time to the most recent common ancestor. Okay, questions? Does that kind of make sense? Okay, so how can we use this to find the root? In the same way that we used in maximum likelihood phylogenetic inference, we had a scoring method that we tried to look over a bunch of different tree topologies and branch lengths to maximize that score. Here, I could theoretically do this process across all possible roots in my tree. So I have a linear number of branches on my tree, right? If I have n leaves, that means that I have n minus 1 internal nodes. Going back from CSE 12, maybe you don't remember that, but either way, we have a linear number of overall nodes in my tree if I have n leaves. And as a result, I have a linear number of branches. So I could try literally every single branch in my tree, find what is the point that is the best, assuming that I root at that branch, which I could do just using closed form arithmetic. And for each of those potential roots, I could just see what is the variation around this line. If I get a super big outlier in one of the root positions, that's a bad option. So I'm basically finding the root where if I root along that branch and then pick the best position in that branch, I minimize deviation from this line. Hello. Does that kind of make sense? So we can estimate the times and the mutation rate just given this plot, and we can do this for every single possible root to figure out what is the best root. Okay. So some tools that exist for doing this. One popular tool is called Tree Time. I think I'm gonna have you run this in class on the lab. So Tree Time actually not only does tree rooting and tree dating, but it also does a third thing called ancestral state reconstruction. So way back in the beginning, I mentioned that true evolution we can think of as like mutations being dropped along the branches of our tree. But when we inferred the phylogeny, we don't know those internal node sequences. Ancestral state reconstruction is an approach for inferring the most likely sequence at each of those internal nodes. So if I do ancestral state reconstruction, I can actually see where did each individual mutation fall along the branches, and also what is the sequence of the common ancestor of all these sequences. There's a question, would this get confounded if the virus goes dormant and doesn't mutate for a while? Great question. So if a virus goes dormant and doesn't mutate for a while, what that would look like is an outlier on this figure, where you're going to have a really late date that has relatively few mutations from the ancestor. And this actually happened with I think, what was it, like 1918 influenza that was like frozen or something like that, and then like re-thawed later on or something. There's like some historical context where sometimes this does actually happen, where like a lab that has a frozen old virus, like it accidentally gets thawed, and like then you have this weird anomaly where it has basically no mutations, but it's very rare. So if you're studying like an ongoing epidemic as it's spreading, you're not going to expect that to happen. The only way that like a virus will truly go dormant and not mutate is if it's frozen like physically. There's weird things with HIV where it sometimes reverts back to ancestral states, but you don't have to worry about that. Okay, so yeah, that's tree time real quick. I know we're at time. Another popular tool is called LSD2. Another one is called Chronumental. It's super, 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 super fast. Basically, each of these tools is doing some type of tree rooting and dating using different assumptions for each. So for LSD2, you're going to be using outgroup rooting in this lab, but it also supports other rooting methods. Tree time, you're going to do the molecular clock approach. I think Chronumental, I don't think I have you run it, but basically a lot of tools exist. Each of them makes different model assumptions and takes different approaches for what it's optimizing to try to figure out the root and the mutation rates. Okay, that's my last slide. Thanks so much, folks. So yeah, lab 7 is out. Please come to lab section to get help if you want it. And I guess I'm done with slides for this week. So think about what you want me to do next time, and I will do whatever you all want me to do. Awesome. Cool. Thanks, folks. Let's see on Zoom. Yeah, thank you so much, folks. Any questions from folks on Zoom before I end the call? Feel free to type in the chat. Feel free to raise your hand if you want to unmute. Awesome. So I'm going to go ahead and end the Zoom call. If you think of anything that you want me to do on Wednesday, feel free to post on Piazza, and I'm happy to do whatever.